Hello, everybody. My name is Roman Bauer, and I'm the spokesperson of the BioDynamo collaboration. I would like to here briefly summarize a recent study of ours where we present the BioDynamo software. So I'm a computational biologist, and as a computational biologist, I'm interested in modeling systems, biological systems that are based on cells. But in order to reproduce those systems, I'm interested particularly in modeling how cells behave in an external environment. And this is actually fundamental to most biological systems. So usually one wants to model some process of how a cell behaves. And also one needs to take into account its local environment, which can comprise, for example, mechanical interactions or substances that diffuse or electrical activity. If one uses an agent-based modeling approach, then one usually models the cells as autonomous agents that can interact with one another and that follow rules that are genetic-like encoded. And this is in contrast to other modeling approaches where there is a global orchestrator that tells the cells what they should do. So to give an example of this, since we have worked for more than a decade now on this kind of computational models, here we see an in vitro system that is simulated, where we start with randomly located neurons, and these neurons grow out neurites in a biologically plausible way. And this model then produces a very complex neuronal network structure based on relatively simple growth rules. And we could actually show that this network structure reproduces many features of the connectivity in cat visual cortex. In another more recent study, we used computational agent-based modeling to model the development of the layers in cortex. And without going too much into detail here, I would like to refer the interested audience to have a look at this paper that was recently published in Cerebral Cortex. Now, these kind of models are very detailed and powerful, but they're also very demanding from a computational point of view. And so in 2014, I reached out to CERN because of their expertise in high-performance computing, and we created a collaboration. And this collaboration, the BioDynamo collaboration, has now grown to a large consortium that includes more than 8 institutions from various different countries. And we have also succeeded in creating such a software that can model complex biological systems in a very performing manner. So we can model, for example, 3D tumor growth, or we can model large-scale neural tissues. And this also means that we can not only model round elements such as cell bodies, but we can also model elongated structures such as, for example, for neurites or vasculature. Our study was then recently published, and we provide also as open source the software and various code examples. And this study was then also recently featured in a research highlight in the journal Nature Computational Science. Now, when one wants to model such an agent-based system, then one has to use certain types of fundamental rules. And in BioDynamo, we have a certain set of potential rules that the users can employ and put together in order to create his model. And one of these behaviors is the move behavior. So this specifies that the element, whatever the element is, can move in 3D space. Or we have the divide behavior. Proliferation is, of course, very important during biological growth and development. We have then also the grow and shrink behaviors, so the morphology changes. We have the secrete and detect behaviors. So, for example, substances can be secreted and then sensed by other cells. The die behavior is fundamental during biological development, apoptosis and cell death. And then also link. So this means that cells can connect to one another physically and create a bond, a physical bond. We have used this framework then for some basic use cases to illustrate its potential and power. In this case, we have used it to model in vitro cancer growth, where we could reproduce the dynamics of the size of the cancer as measured in vitro using different initial conditions. And as you can see here, it matched very well. Then also we published work where we used BioDynamo to model large-scale cancerous growth. And also here we could model, for example, how the oxygen distribution changes over time, or the density of cancerous cells in 3D space. Then we have also modeled individual neuronal morphologies with high detail. And in this case, we have actually a visualization of a simulated neuron as well as one of an experimentally measured neuron. And I leave it to you to decide which one is which. And feel free to reach out to me if you really want to know the answer. And recently, we also started using BioDynamo to model large-scale organs, because we are interested in optimizing the cryopreservation of organs for biomedical applications. We are very interested in connecting with other research groups and researchers in the computational biology community. And we have created a website where interested people can inform themselves. And importantly, there are also many demos. So you can have a look at the different, a large number of use cases, and also you can try the code out yourself in the browser. So you don't even need to install anything because of the Jupyter Notebook integration. Ultimately, we would like to use this approach for large and complex systems, such as, for example, entire brains. And to do this, we will make use of the principle of biological development, where the complexity actually increases exponentially from a relatively simple setting. So the human brain, for instance, has almost 100 billion neurons, but it develops from the genetic code, which has only approximately 20,000 genes. And so we want to employ this nature-inspired approach also to model really complex biological systems. I would like to also highlight that we have been engaging in many outreach activities and events, and we plan to do so or more in the future even. And so if you're interested in BioDynamo or would like to learn more, then please do not hesitate to contact us, either via contact@biodynamo.org, or you can also directly contact me at the University of Surrey via r.bauer@surrey.ac.uk. And of course, you are very welcome also to follow our Twitter account. Thank you.